Hello. In the last lesson we talked about how nerve cells or neurons function, and specifically that they have the ability to conduct electricity or conduct an electric signal. So they can send a signal from one cell to the other. Specifically, they can receive the signal up here in the dendrite. So the signal will come usually from another nerve cell. It gets taken up here through the dendrite. And it might in fact take multiple signals from multiple cells, so you might have several signals coming in, and then all these signals get combined at the axon hillock here, and then basically the cell decides whether or not it sends the signal onwards. Now if it does, it basically will conduct the signal along the axon, which is the long part of the nerve cell, to the base of it, until it reaches another synapse here, which is the connection to another nerve cell or potentially also another muscle cell. These axons can be very long, they can be over 1 meter, but really there is only one axon, but on both sides we have branches at the top. We have potentially several hundreds or even thousands of dendrites, so lots of different places where signals can come in, and then also it can form several different synapses to other cells, so it can connect its signal to many different cells. Now really for this to work, the cell in fact needs to create an electric potential, so it makes an electric current. Now when we talk about an electric current, really what we're talking about is the difference between the electricity inside, or the electric charge on the inside and on the outside. So basically the difference between the inside and the outside here is what we talk about. Now normally, if the cell is in its what we call resting potential, this difference is in fact minus 70 millivolts. Now, why this is, how we get to these minus 70 millivolts, is something that I would like to explore a bit further, because this is really a chemical property of the cell in order to get to this so-called resting potential. I'm sure you remember from last year biology that all cells essentially have membranes, and it's no different here. So we've got the normal cell membrane here, made from phospholipids. So we're talking about the inside of the cell. In this case, that would be on top here. So here's the inside. And then obviously on the other side, we would have the outside of the cell. Now the real reason that there is a difference in electricity is, nice, maybe to repeat, just as I said before, what we want to explain is why on the inside we've got a negative charge, and therefore on the outside we've got a positive charge. So the real reason that there is an electric difference is because of different ions, and two ions matter especially in this context, and these are sodium and potassium, because they are in fact in different locations. The sodium is mostly on the outside of the cell, so we got plenty of sodium out here. Here, plenty of sodium on the outside. Now some sodium will also be on the inside, but by and large this is mostly on the outside of the cell. So you should remember from chemistry class, sodium is positively charged, Na+, sodium, the German natrium leads to the Na+. And on the other hand, we've also got potassium, which is what matters here, and potassium, unlike sodium, is mostly on the inside of the cell. So we've got plenty of potassium ions which sit on the inside, not on the outside. and these are also positively charged. So that in itself is not enough to explain the difference. We'll have some potassium also on the outside, but as said, the majority of which sits on the inside of the cell. And the potassium, in German Kalium, leads to the chemical symbol K. Again, these are both positively charged, so both of them carry a positive signal. Now on top of that, how do we get to the negative charge? There are other ions that matter. In particular, we've also got chlorine, so Cl-, and the chlorine again is mostly on the outside, so we few little chlorine ions. They don't actually end up moving that much, but they are responsible for creating part of the charge. So we got Cl-, chlorine, but then almost most importantly, we've got plenty of proteins, so large protein cells, and these proteins are by and large on the inside. Again, the green color here. So the proteins, they are very large, and that's the reason that they basically cannot come out. Maybe use a larger pen here. So we've got these big, large, chunky proteins, and proteins are almost always negatively charged, which is the reason we have an overall negative charge on the inside. So all these different chemical components contribute to the charge, all differently, but not all of them can change it actively. Particularly the proteins, they essentially stay where they are, and therefore they don't sort of contribute to the changing potential. So we've got proteins here, and proteins are always negatively charged. Right, so that in itself explains how this charge comes to be, but then importantly, these things can move, at least some of them, but they can't really move directly through the membrane because the membrane will block almost anything. So what we need are different membrane proteins. So we've got specific proteins for both sodium and potassium, which allows these ions to enter and leave the cell. So in effect, we've got potassium channels, which means that the potassium ions can leave the cell because they're mostly on the inside. So that means if they want to, they can go here through these channels and exit to the outside. On top of that, we've also got specific sodium channels, and these sodium channels allow the sodium ions, which are mostly on the outside, to enter. So they can come in. Now there are two reasons why any specific chemical might want to enter or leave the cell. So there are two factors that we have to consider, and these factors let's zoom out a little bit. Factors that influence ions. These factors are essentially the concentration that we already have. So the more ions there are, because of diffusion, they want to move to a different location. But also, so one is concentration, and the other one is charge. If an ion is positively charged, it obviously wants to go to an area of negative charge, and vice versa. If an ion is negatively charged, then it is much happier in a place where there is positive charge already. So what does this mean? This means specifically that at the moment the sodium and the potassium are the ones that can move in and out, but they are affected differently depending on what's going on around them. To be specific, the sodium and the potassium ions are affected differently. If we look at the sodium first, the sodium is currently mostly on the outside here. Mostly on the outside here. The concentration is fairly high, so due to the concentration, the sodium would prefer to come in. But the charge is positive, so as well, because there is a positive charge and the sodium itself is positive, they are also driven in. So they've got two factors that are driving them to the inside. Now, as a matter of fact, they would quite like to enter the cell, but they cannot, because these sodium channels here, they are not open. They are in the standard resting potential. They remain closed. So that means the ions would quite like to enter the cell, but it is impossible for them to do so because they get blocked by the membrane. If we look at the potassium on the other hand, the potassium, because of the concentration, there's lots of potassium on the inside and very little on the outside. So the concentration would in fact be driving them out because they would like to disperse. But inside it is nice and negative, mostly because of these proteins up here that keep a negative charge. And the potassium itself is positive. So that means due to the concentration, they would rather stay inside. So they more or less, these two factors neutralize. That means even though the potassium has an easier way, these channels are not completely closed, but some will in fact permanently leave and others will come back. So these factors roughly keep in check, which is responsible for the fact that there is more potassium on the inside and less on the outside. Now, what would happen over time if we didn't actually affect this is that slowly some potassium would start drifting out. We call this a leak current. So we have overall a few more of these potassium ions that end up on the outside here. And as they leave, the charge of the cell starts to drift, and in fact it starts to go more towards the positive on the outside and even more negative on the inside. So as said before, if we just left this cell alone, it would slowly start to drift, because even though the sodium is mostly blocked, it is not perfectly blocked, which means over time we'd have a slow, what we call a leak current, that some little sodium ions would still manage to sneak past here and get into the cell. And if this happens, they will bring in more positive charge from the outside, so we start to see more positive charge out here, and obviously then we start to see the outside charge go more towards the neutral or towards the negative. So very slowly the difference of the cell would start to drift away from those minus 70 millivolts that we said was the difference. So really the minus 70 is what needs to be kept up. And as soon as the sodium starts to drift in and the inside becomes more positive, then also the potassium is no longer kept in, and this would also start to drift out. So in order to keep the cell happy, it actually needs to do some work, and for that we have a third ion channel, and this is what we call a sodium-potassium pump. And you can see that here, this has the ability to pump the sodium back out that has managed to sneak in, and it also has the ability to pump the potassium back in. So essentially, the sodium-potassium pump, you can see it here, it can pump, take it from this side, it can pump 2 sodium ions back out if they've managed to sneak in, and at the same time it can pump 3 potassium ions back in order to keep the system stable. So this sodium potassium pump will actively move the potassium in and the sodium out. Now, nothing of this is free, so the sodium potassium pump has to actively move the ions from one side to the other. So this in fact uses energy, and energy in biology, as always, is in the form of ATP. So what this means is the resting potential has to be constantly maintained by spending ATP. So in order for the potassium to stay in and the sodium to stay out, the cell needs to constantly expand energy, and if it stops doing that, the potential here, the minus 70 millivolts, would start to drift. So we can only keep those minus 70 millivolts if we continuously spend more and more ATP. So that means even though your nerve cell might be addressed and you're not doing anything, you are in fact constantly spending energy. So just to keep the system happy, energy is constantly spent. Energy constantly spent. So that means even though you're not doing anything, just in order to keep your nervous system happy and ready to actually process some information should it ever arrive, you are spending plenty of ATP. And that explains why our brains are so costly, because even though when we're not using them at all, just in order to keep them ready and happy, we are spending a lot of energy. So our nervous system, and particularly the brain, uses about 20 to 25% of our daily energy. Even though we're not doing anything at all potentially, we are still using that energy. So the nervous system is one of the most costly systems in our body, and the only reason this pays off is because it gains such a massive benefit, because we can do algebra and communicate and build rockets and run entire economies. So that is the reason we still keep this energy upkeep, but plenty of animals do not, and they are quite happy. The brain really is the costliest thing you can run in your body, and the resting potential explains why that is, because we constantly need to keep the system fed with more ATP just to have it ready for when potentially we do want to send an information. And that sending of information is what we call the action potential. So that's when the cell will actively send a bit, a piece of information, and really what it does, it flips the negative charge to a positive, and how that works we will look at in the next video.